Okay. So while we wait for media services to show up, same thing happened this morning. So they were able to fix it within a few minutes what they got here. We're going to dry run it pretty much, fly blind a little bit. So we're talking about mitosis, right? So that's chapter 10. And one of the things we wanted to talk about, we talked a little bit about chromatin structure. If you recall, we had the little histone complexes, right? The little drums around which you had DNA wound around it to basically kind of stabilize the DNA. We talked a little bit also last time about it looks like the same thing. The little screen is black again and doesn't seem to respond. So it's, that's fine, yeah. That's fine. I can just re-jump on there because this is the main one. So basically, we talked a little bit about heterochromatin, right? So basically, when you take a look at chromatin structure, you have your little chromatin drums, your little histone drums, and around which you have your DNA wound around that. And we talked a little bit about the chromatin structure. We talked a little bit about heterochromatin, where you can take that pack and you can pack even tighter. And when you do that, what it does is it creates inaccessibility for any genes that have genes. So it's kind of a way for you to actually silence your genes. So we learned that there's a lot of chromatin remodeling associated with just gene silencing and gene regulation, which is actually a fairly recent discovery. We used to think that there's like proteins turning things on and turning things off and stuff like that. And as it turns out, we have all these chromatin structural sorts of things that are creating inaccessibility. That's a big deal for us. And so when you take a look then at this little chromatin fiber, what you can do is you can basically wind this around in sort of like a little coil. So you can kind of wind this around into like this little coil. And the diameter of this is about 30 nanometers. And so we tend to call this either the solenoid, because it's like a little spring, or we call it the 30 nanometer fiber. I tend to call it the 30 nanometer fiber because that gives you some sense of its dimensions and how big it actually is. And so it's this little coil of chromatin. This is like what it looks like in a normally non-dividing cell. But when you actually have to go to divide the cell, and this kind of gives you a little bit of range, right? So you can go from here, or you can pack your DNA tighter, or you can loosen it all up. And so it all depends on whether or not you need access to like a little gene that's right there. Because remember, the DNA is a working molecule. It's not just like a dusty old book that just sits on a shelf somewhere and nobody ever reads it, right? It's actually a working document. So you have to contact it a lot. You have to work from it. You have to build from it. You have to know which amino acids to pick when you're building your protein. So you need access to that gene because that gene is going to tell you which amino acids to pick. And so you're not making this protein all the time, right? You're making this protein at certain times in the cell's life, and at certain times you don't need it at all. So when you don't need it, it's okay if you kind of bundle it all up and package it all up and sort of keep it off for safekeeping. But when you actually have to get access to it, you want to be able to also get to it and open it up so you can read it, right? So you want to be able to kind of go back and forth a little bit. So you have this need both for structure and also for accessibility. And you can't have both at the same time. So you're kind of always playing sort of balancing game between these two. But you have this dynamic range, right, between the 30 nanometer fiber and the basic, which is the most accessible version of the DNA itself. And this here all, and like a normal cell that's not dividing, you're in various states of remodeling, which is the reason why chromatin remodeling research is like so active, because we're trying to understand exactly like in the life of a normal cell, like what does your remodeling look like? Well, how do you know when to go to the solenoid, and how do you know when to loosen up, and how do you know when to package down? Who controls that, right? Who drives that? Who's the managers of that? So there's like all these questions we have about this basic process and how it kind of moves around. And so there's a lot of research going into that. And a lot of people over at Anschutz who are studying a lot of that. And so this is basically, your solenoid. But when you actually go to divide them, you have to actually condense stuff down. So think about a dividing cell and your chromosomes. Think of it as sort of like you're moving. Let's say you're an East Coast family. Let's say you're a family that's been there for generations somewhere on the East Coast, and you've got this fine china collection. And it's a collection that's been in your family for generations that goes way back. And each generation is added to it. And so it's a really kind of an invaluable sort of a collection. And it's all on display, right? You got the china hutches. You almost never use it. You don't do stuff like that. You just look at it, which kind of defeats the purpose, right? The idea of plates is to eat off of them. So if you're not eating off of them, what really is it? You just go buy a picture hanging on the wall. But anyway, then what you have to do then is if you plan on moving from the East Coast to say LA, there's an entire process of moving that, right? So you want to move your china collection. How do you do it? Well, you do it very carefully, right? Because you know there's no replacing this if it breaks. So think about the steps that you do to move this fine china collection, right? I mean, you do something probably like a lot of people do, right? You just basically take the plate and just give it to any random guy on the street and say, here, take this alley for me. No, what you do is you take it all off of display, right? You probably organize it all. Grab the plates over here on one side, the cups and the saucers over here, right? So the different types of things are all kind of in their little places. And then like when you go to pack the plates, you don't just pack them on top of each other. You put like some sort of little padding in between them, right? So there's no clinking going on there, right? So one plate is never touching another. Same thing's also true for all the rest of your dishes. And then you put those in a box. Thanks a lot. So you put them everything in a box, right? And you fill it all in with what? More packing, right? Packing popcorn. And the idea is you probably pack it so tight that nothing is moving in there. I mean, you could probably throw this thing off of a cliff and it will never budge. You've got this thing packed so tight, right? It will not move. But then you sort of wrap it all up with the heavy duty packing tape and you ride all over it, things like fragile, right? Children's children for 10 generations. That's a little extreme. But you know what I'm talking about, right? If you've ever broken anything valuable during a move or lost anything valuable during your move, it's like, oh my gosh, I'm never going to live that one down, right? So yes, you will kill people for 10 generations. So what you do then is you just give that one to the mover. Yeah, here, take that box right there, the one with all the death threats on it, and take that one to LA for me. Do you even do that? No. Who's moving that box? You, right? You're like, no. Get your greasy paws off it. This one's mine. I'm going to tuck this one under my wing. This goes with me. I sleep with it, right? It is in bed with me. I kind of pull up the covers over it, right? Kiss it goodnight. Give it a little bedtime story, maybe. Read to it. Okay, maybe not quite that extreme. But it depends, right? It also depends on how your mental health is. I mean, if you've ever gone through a move, your mental health could be there, right? Right. It's kind of like Castaway. It's Wilson for you, right? So it's not longer a volleyball anymore. It's your friend. So then you get to LA and everything's perfect. And then what do? What's the first thing you do when you get to LA and you get into the new house?
Get it to a safe place.
Yeah, right. You find those china hutches, the display cases and things like that. And the first thing you do is you go after that china collection and you pull it all out of the packing and you put it on display. And it's like before you even have like a bed to sleep in, that's the first thing that goes up, right? Notice what you've done though. What you've done is in order to move your china around, what you've done is you've taken your collection and you've condensed it down into a small movable package. And then once you've done the moving, you've decondensed it, you've taken it apart and you've put it back into normal operation. You're doing the exact same thing when you go through cell division. the first thing you do is you pack it all down into a safe, movable package. That's called chromosome condensation. Once you move the chromosomes then safely and they get to their final destination, then you unpack it and you put it back into normal operation. That's called decondensation, chromosome decondensation. And those are some major activities, some major points that are associated with this process of moving. So let's take a look at some of this condensation, what's going on here. So we started off, first of all, with our solenoid, right? So this is our 30 nanometer fiber. And so you can see how basically you just kind of wind the little chromatin fibers up into this nice little coil, this little 30 nanometer diameter coil. Now, when you want to condense it down, what you do is you take the solenoid and you basically cap it with some scaffold proteins, creating a hairpin. But you don't stop there. You take each of these hairpins and you make several of them. And you grab like a long sort of a rod shaft-like scaffold protein. And then what you do is you use these little tips, these little scaffold protein tips of the hairpin, and you plug those into the rod. So you basically just plug each one of these little hairpins into the rod with those little scaffold proteins. And you line them all up. And what you create then is what's called a rosette. And you do this for as much DNA as you've got. Now, if I were to take this particular rosette situation and turn it on its side, so you're looking at the rosette's edge on, then it would look something like this. So I'd see basically my central shaft protein, and then I would see a rosette, the edge of the rosette. But then I'd pack another rosette right behind it, and another one right behind that, and another one right behind that, and so forth. And then I'd pack as many. And some of these are going to be big, and some of these are going to be smaller. They're going to be different sizes. But it's going to start looking something like that. So if you kind of follow the outline, it starts to look a little bit like a chromosome, which is the cheese puff stage, right? So that's basically all of your chromatin condensed down into these rosettes on these shafts, and you basically line them all up on these little chromosomal arms. This here is your mitotic chromosome. This is the one that you're ready to move. This is your fine china, all packaged and padded and bundled up in its box. And now you set it underneath your wing, right? So that you can move it. So this is the chromosome condensation process. So that kind of leads us to karyotypes. You already know about karyotypes because you guys actually did a karyotype before break. So when you did that nifty little fourth grade kind of cutting everything out, drove you crazy a little bit. So that's a karyotype, right? So anytime you're taking the chromosomes and you're all lining them up, right? Ones with ones and twos with twos and threes with threes, and you line them all up 1 through 22, and then the X chromosome, the Y chromosome. That's a karyotype. And so it's the best way actually to show you what we mean when we come to the next couple of terms, which is the idea of ploidy. Ploidy basically means chromosome content. So ploidy is a root term for chromosome. And we have two different things. Some organisms have different types of ploidies. What that means is they have different numbers of chromosomes. So for instance, humans are diploid, right? Di-2-ploid chromosomes. And what this means is you have 2 of every chromosome. And so we usually abbreviate that as 2N, where N equals the number of chromosomes in the genome. So we have 23 chromosomes in the genome. We have 2 of each. That brings us to 46. Okay. So the alternative to that then is going to be what we refer to as the haploid. Haploid basically means you only have 1 set of chromosomes. So that's the N. So we've already defined N. This is also, and I like to define it this way. because this is kind of more the genetically accurate way of defining it. Typically speaking, if we say that an organism has a haploid number of 23, what we're saying is that the organism has 23 unique chromosomes, right? That doesn't mean that it has 23 chromosomes in each one of their nuclei, right? That just means they have 23 different chromosomes. They could have 2 of each, they could have 3 of each, they could have 4 of each. So some organisms can be triploids, 3 sets of everything. Some can be tetraploids, 4 sets of everything. And so this is something that's actually fairly common, right? So we have 23 unique chromosomes and we have 2 of each. So we have, now, the other thing also that we have is just to kind of give you a sense of ploidy, kind of where you run into this. I mean, because you probably don't think about it a whole lot. You think it's like a diploid, a haploid. These two terms are actually quite important because whenever you're looking at any kind of an organism anywhere, the life cycle the organism lives is going to be spent in some sort of a kind of a biological reality, right? So the organism actually either lives in a diploid body or it lives in a haploid body or a triploid body. So this is kind of like the reality of what your body is. Your body, what you see in the mirror as your primary body, is diploid. So all of your body cells are diploid. So if you were to take a biopsy of any tissue in your body, except for red blood cells, because they don't have nuclei, but any tissue, you're going to see 46 chromosomes in your nucleus. That's what your reality is. For you, your body reality is as a diploid. But there are some organisms where they actually have a haploid body, which would be weird for us because we don't spend time. We only have actually 2 cells that are in the haploid state in our entire body. And actually, we don't have them all in the same body, right? That's our sex cells. Males, the only haploid cell in a male is the sperm. And in females, the only haploid cell in the female is the egg. Everything else is diploid. So every organism has a life identity associated with their state, their diploid or haploid state. And not only that, but genetically, we've kind of messed with this a little bit. Now, here's a good example. How many of you guys like strawberries? Yeah, I love strawberries. One of my favorites. Bananas and strawberries are like the best combination on the face of the planet. It's hard to beat. Maybe if you toss mango and pineapple, that's a pretty close one. But traditional, oh my God, I'm a sucker for the classics, right? I mean, we all start off eating banana puree and we never stop, right? Unless you hate bananas, but that's the thing. But a good strawberry, right? You go to the store and what strawberries do you look for? You look for those big old bright red monsters, right? But here's the problem. Have you ever seen a wild strawberry? Oh, I bet you some of you have. I've seen them in Denver. They're almost imperceptible. You probably don't even know you're stepping on them. If you look at them, they're like tiny, tiny, tiny. The biggest one is like maybe the size of my pinky nail. They're very tiny. Yeah, and when you look at them, it's like it almost seems like Gulliver's Travels. It's like, the Lilliputian strawberry or something like that. It's like, wait a minute, is that you're looking at it, it's like, something looks familiar about that. And it's like, and you look at it, it's like, oh my gosh, is that a strawberry? It's like, it's so tiny. It's like, it's a little bitty thing. And that's a diploid strawberry. That's like a wild strawberry. Obviously, that is not the strawberry you guys buy at the store. So what we've actually done, and this is actually true for most produce in the store, what we've actually done is we've jacked with them. So pretty much everything you buy and eat has been modified. Nothing is natural. Nothing. So the way we get those strawberries to be so big is instead of being diploids, they're like octoploids. So we've genetically forced them and manipulated them to carry 8 sets of everything. And that makes them bigger. There's just a correlation between size and the number of chromosomes you have for plants, not for us. It's lethal for us, but for them, they can do that.
Does the only effect that it does is make them bigger, or when we ingest a plant with all those chromosomes, does it affect us worse than?
No,
So it's really not a problem.
No, we're just eating DNA.
So it's
So-
a good thing, because we can make food.
Eat more
bigger.
strawberry. Yeah. I mean, it's not a bad thing. What?
I just-
Yeah,
I feel
it's a productivity. It's definitely a productivity boom for us, right? So, but it's kind of, I find that historic bananas the same way, right? We talked about bananas. I mentioned bananas for a reason, not because they go well with strawberries, but because, have you ever noticed, when you chomp into a banana, are there a bunch of seeds in there?
Yeah.
Does that kind of confuse you a little bit? Because what's the purpose of a fruit? Yeah, so where are the seeds in the banana? A wild banana has seeds in it. A wild diploid banana has seeds in it. The banana that we see in the store is like a tetraploid. So we've actually genetically altered it to lose the seed, just like we've done with seedless watermelon. So like I say, literally nothing you buy in the store has not been modified to some extent. In some cases it's mutation. It's a form of a sterile, kind of a sterile thing. Like sometimes when you make like the donkey, like the horse and the donkey makes a sterile mule, right? So sometimes when you do that, it'll create the sterile offspring. And that's for a plant, that would be like a seedless version of it. So you can do something like that.
Sounds like selective breeding of the plants.
That's exactly what it is. It wasn't just with animals, it was with pretty much everything. But everything's been modified. There's nothing that you buy in a grocery store that hasn't been modified, which is kind of the reason why the whole GMO thing kind of sometimes smacks a little comical to me, especially watching people in the store get all uptight about stuff. And I'm like, so you're kind of upset about this thing over here, but you're willing to eat the monstrous strawberry. So does that not bother you? I mean, strawberries don't come that big unless you've jacked with them. I mean, so apparently, right, where do you draw the line, right? So, because everything's been modified. So delineate what acceptable modification is and what unacceptable modification is. And most of the GMO proponents don't have that. They just say, oh, it's been modified. Well, guess what? You're not going to get something that hasn't been modified. So I hate to burst your bubble, but you're going to have to starve to death if you really want to take a non-GMO stance. That's a hard line.
I think we just need to be more informed on like what is bad GMOs and good GMOs. Most
And-
people have more.
Well, and most people who are actually proponents of it don't have any idea because they just know it's like, oh, well, I don't like the idea of that. Well, why? So I don't know. I just don't like the idea of that. It's like, okay, that's not good enough, right? You need to have a reasonable reason why that's not good enough, because I may be okay with strawberries. There may be somebody out there who's now looking at strawberries thinking, I can't eat you now because you've been modified. Well, okay, that's your choice. I'll just enjoy the strawberries. I'll just buy more. But I mean, different people are going to come down differently. But the problem is there's a lot of undevelopment on that. There's a lot of heat and passion on that side, but not a lot of brain involved in it. Which is if you catch that pattern, that's the same type of combination that leads to things like witch trials and burning people at the stake. Right. Welcome to the species of lemmings, better known as Homo sapien lemmings. Right. We are not as evolved as we think we are. Oh, you mean like for CRISPR and things like that? Oh yeah, that's red hot. And there's good reason for that, because that's exactly where that goes. And I think there's a lot of ethical questions associated with that. And people have a right to be upset about that one, right? So the thing about technology, and this is kind of in society, this is like a little sociology 101, is, when you envision a future where you have designer babies, and honestly, let's face it, I mean, the reason why people don't like the idea of designer babies is because it smacks of Adolf Hitler. I mean, essentially, you start devolving into things like master race. So you're making choices against one race in favor of another. It becomes very racially divided. when you start going for designer babies. Because just by your consumer approach, you're essentially picking and you're deciding who is worthy and who is not. That goes nowhere good except for where the Nazis went with it, only in a high-tech version of it. So most people are kind of repel against that. But let me tell you something. It is the slippery slope. And if you're too socially narcoleptic, I would suggest you wake up. Because the way those sort of things, the way you wake up in a future where you have designer babies and massive injustices and people being denigrated to second-class citizenry and maybe even organ farming, you don't just wake up thinking, hey, you know, I'm going to create this horrible world and I'm going to make all these decisions and we're just going to just create this hideous nightmare of a future. Nobody wakes up that way. It always starts off as a great idea. CRISPR is a perfect example, because the great idea of CRISPR is, well, wait a minute, what about this genetic disease this person has? I mean, who wouldn't want to cure them of this genetic disease that they have no hope of getting over? I mean, think about it. What monster would say no to a person who could potentially have this life-saving CRISPR technique done on them, and you could change their life and you could save their life? Wouldn't want to do that. Well, that sounds reasonable, doesn't it? A lot of people aren't going to push back on that one because nobody wants to be called a monster, right? I mean, most society is crippled by teenage level peer pressure. Sorry, that's just, go through a political environment, go through a couple of elections, right? We're all just a bunch of insecure teenagers and nobody wants to be on the bad side, right? Everybody wants to be on the acceptable side. I want to be on the winning team. So you'll do whatever the winning team says is right. That's just the way humans are. So who wouldn't do that, right? Because you can imagine just the horrible smear campaign you would get if you said, no, we're not going to do CRISPR. I don't care if it is saving this person's life. Can you imagine just the absolute character annihilation that you would go through if you said no to that? But guess what? You say yes to that because most people aren't strong enough to say no. And then that's where you start. You stepped out on the icy slope. And eventually you'll be like, well, yeah, but you know, this one's not life-threatening, but you know, this should be okay, right? I mean, oh, okay. I mean, it's not life-threatening condition, but okay. You know, we'll change this one. Oh, but this one's okay, isn't it? I mean, that's not really a disease at all. It's just kind of, you know, hair color or something like that. You know, I mean, that's okay, right? I mean, we're not saying anything. We're just changing hair color. I mean, we're not like being evil here. I mean, yeah, right. So like, and it slowly you slip. And for the most part, that's the way change happens in America.
I think it's snowball.
Yeah, you ask any political activist on their most cynical days when they're like open and honest, not in their normal deceitful lying way. If they're honest with you, they know full well the psychology of change in America. You start off with a flamingly repulsive idea. And then you don't just sit out there and say like, here, sign this bill. It's incredibly repulsive. Of course, nobody's going to sign it. What you do is you start the war of attrition. You start setting up your network for generations. Okay, when I'm done and gone, you pick it up and you continue fighting. And you chip away and chip away and chip away and chip away. And that's the slippery slope effect. Unfortunately, that's the reality of how America works. So when we get to the future that we all hate, we only have ourselves to blame. Wow, that's pretty cynical. But I've been alive in this country long enough to see it happen in real time. Sometimes for good, but sometimes for bad. Yeah. And any political strategist, if they're honest, will tell you that's exactly their game plan. Exactly their game plan. It's the reason why I have no respect for politicians. That's why, for me, elections are some of the worst moments in life. Because it's like, wow, you're voting for people who aren't even human. to govern a bunch of people who are human and struggling to be even more humane as humans. So you're kind of like throwing the pearls to swine, basically. That's pretty off the charts there, but that was all free, by the way. There won't be any exam questions on that one, so don't worry about that, right? But that's just a little shoot from the hip, from this side of the eyeballs, so to speak. So, but anyway, used to be back in the day, that's what college is for, right? Kind of talking our way through all of the realities of life and how to change the world. But now we've kind of turned into more of a training ground for skills and crass job looking stuff, and not really character development anymore. Self-discovery, those sorts of things are largely been destroyed by the very same people who created them in the first place, our lovely boomers. What?
To beat the game, you have to play the game.
And that's true. And unfortunately, and when you play the game, you oftentimes become the game. And then usually you get gamed. So you become institutionalized. Yeah, that's what happens. It kind of reminds me of one of my favorite Star Trek episodes, the one where Picard was taken over by that little, they created like an alter ego for him, like a personality for him called Cayman. And he was like on this dying planet where the sun was getting ready to consume the planet. And it was like their last beacon to let people remember this dead civilization. And he was fighting it at the beginning. And then eventually he kind of settled into his role as Cayman, and it kind of showed him how he got married and he had a daughter, and his daughter was a science nerd like he was. And he was kind of like going through this whole life of this guy called Cayman. And then it turned out to be like, that was just like their little, remember us, we're not here anymore because our sun blew up on us, but remember us. It was one of my favorite episodes, but that's kind of what it is. Like the more he fought it, the more it kind of entrenched, and then eventually became institutionalized, and then he didn't want to leave. So it was kind of funny, but yeah, that happened. So when you take a look back to normal stuff, science stuff, so let's a little diversion there. So, but then when you take a look, and we can suffer that because we're way ahead in the schedule. We're doing quite well actually in the schedule. So when you have these chromosomes, right, you have the mom chromosome and the dad chromosome, then we refer to them as homologs. Why? Because they're same, but different. What does that mean? Right, it's an oxymoron, right? How can it be same and different at the same time? Well, it's the same because when you take a look at mom one and dad one, they both have the same genes, but they have different forms. Let's take hair color, for instance, right? So on a chromosome, dad may have the hair color gene, right? And his hair may be black. And he comes from a long family of whatever you want to call it, but that's like everybody in the family has black hair, which is probably describes the good majority of people in the world, by the way. I'm just letting you know that. Right. So, and then let's say mom has like blonde hair, like bright blonde hair, all the recessive traits, right? So blue-eyed, blonde hair, that's what. So they both have a gene for hair color, yes, but they have different forms. Dad has the dark form, mom has the light form, right? So same, but different. So those are homologs. And so they're important, right? Because they kind of share a sense of variation. So in that individual, if dad had dark hair and mom had light hair, then you're now carrying both the gene for dark hair from dad and light hair from mom. So you've got a combination of the two, even though technically you may look dark, right? But you're carrying the genes for light, thanks to mom. Now, obviously, it's a little more complicated than that with hair color and things like that, right? Because I mean, we do have a lot of like interracial marriages, a lot more now, which is really, actually genetically, it's a really good thing. Because the more we intermarry, the more like there's like just different gene frequencies and different like different forms of genes that like normally these two cultures wouldn't have access to. But when you intermarry, then you get to add new sources of DNA into that population, and that makes everything stronger. That increases variation. So you start to get much more variation, and you can actually see that in an interracial family, right? So like if you have like a father who's really, really dark and a mother who's really, really light, and then have kids that are kind of like all these ranges in between, right? I mean, some will be a little darker like dad, some will be a little lighter like mom. You have some that are more olive colored, and the hair, same thing, right? The curly versus straight, that sort of thing. I mean, you can see all kinds of variations in between. So just having that mixture is a good thing, because that creates more variation in the amount of DNA, and that's always a good thing, having that extra variation. So that's a good thing. So that because we're starting to become a much more progressively interracial society, and like interracial marriages, where I mean, they used to be like, oh no, what? It's like now it's kind of like, yeah, so it's like that's not a big deal, right? I mean, it's kind of like passe. I mean, that was a good thing, right? It was always supposed to be normal. It just was, just society evolution is what it required, right? But that's like a good thing, because not only because I was just talking about physical traits, right? Just the way individuals look and the variation of traits, but even more important, and the reason why that's more important is also with like disease genes, right? Because two different cultures will have a completely separate set of genetic disorders that sort of bedevil them. And so when they intermarry within that culture, they compound those genetic disorders, and they're more frequent and they're more likely to happen. Whereas if they're intermarrying interracially, they're spreading that out and they're sort of complementing each other. So it's like, so it'd be like my disease genes will not be somebody else's disease genes. So if we get together, then we complement each other. So their disease genes are covered by my normal, and my disease genes are covered by their normal. So it strengthens everything. So it's actually a good thing. And it creates an actual. So if you think about it, when you want to really, talk about Hitler and his so-called master race thing, he was actually genetically completely backwards. Really, strengthening a genome requires strengthening variation. So if you really wanted to create the so-called master race, interracial marriage is a better thing to create a master race, because it gives you much more variation, robustness that really eliminates a lot of genetic downfalls when you kind of get siloed in one particular cultural grouping. And some are worse than others. Some groupings are worse than others, because that's just the way we used to do it back in the day. It's like Germans are really bad, right? Because they would like hunker down and they would all intermarry with each other. And then, all these disease genes would just compound in there. But if you start to intermarry, it becomes less frequent. So you eliminate some of those bad genes. And so that's actually a good thing. So that's why I say, all the eugenics people had it completely backwards. But the reason they had it backwards is because it was never about genetics. And for them, it was never about that. It was basically about racism. It was about racism and annihilating races that they didn't like. So it had nothing to do with human decency at all. Inbreeding, yeah, because they would like marry their sisters. Yeah, the pharaohs were terrible. Oh my gosh, they were train wrecks. And one of the reasons, one of the things is like some of the pharaohs, like Tutankhamun, they thought maybe that he was so screwed up because he was so grossly inbred, because back in the day, they used to believe that, royal blood was better than, commoner blood, apparently. So you would intermarry in your own family. So of course, Tutankhamun's wife was his sister. So it doesn't surprise me that they weren't able to have any kids. Actually, they did. They had 2 kids, 2 daughters. Both of them were stillborn. So it's like, yeah, gee, you think. Do what? If they're inbred? Yeah, the reason why inbreeding is bad is because you compound. It's like the ultimate distillation of all of the bad disease genes. So if you've got disease genes floating around in the family, you're going to make it so frequent that you're going to just be like a genetic train wreck. Yeah, it doesn't tend to work very well. And in genetics, it's a term we call outbreeding. So breed with a genetic background that is not your own. And that creates genetic diversity, right? And if you think about it, there's kind of there's for the most part in most mammals a natural compulsion for that, right? We oftentimes, we have a mantra in society, right? Opposites, right? It doesn't say sames attract, right? There's no such thing as that. Why do opposites attract? Well, the reason why we're attracted to opposites is because we're kind of almost instinctually attracted to something that doesn't look like us. Why? Because that's an instinct that would ensure getting away from our own genetic background so we don't inbreed. So we marry into a different family with different genetics, and that's a good thing, right? Unless you're from the South. Sorry, I'm not going to start doing the Southern inbreeding jokes, but
there's a documentary called The Whitakers about a inbred family from West Virginia. It goes in depth and it's pretty bizarre. If anybody wants to check that out.
yeah, it's usually nothing good happens when you start inbreeding. It's just it's a recipe for disaster. Yeah. So remember our mantra for cell cycle, right? There's a 2-step one. The first thing you have to do is duplicate, right? And then you have to separate, right? In that order. And so this is kind of the duplication part of it. Now, we're not going to talk a whole ton about all of this because we got an entire chapter about replication. We'll save a lot of that for then. But ultimately, the first step is your chromosome replication. And so when you take a look at your chromosomes, for the most part, what happens is you start off with your 2 chromosomes, one from mom and one from dad. And you've got basically 2 copies of them, right? So this is your little centromere. And this is before replication. So then once you replicate, then what's going to happen is you're basically going to have the same centromere, but now you're going to have 2 copies of these attached to the same centromere. So these are 2 copies of mom 1. And then you're going to have 2 copies, dad 1. So at this particular point now, how many copies of chromosome 1 do you have? You have 4, right? So you have 1, 2, 3, 4. Each one of these are called sister chromatids. You have 2 sister chromatids attached to each other for mom 1 and 2 sister chromatids attached to each other for dad 1. Okay. Now that's post-replication. And they're held together by this protein that sticks them in the middle between the centromeres called cohesin. So this is like a glue-like protein. That holds them together because you're not ready to separate them yet. You want to organize them a little bit. You want to set them up a little bit, and then you're going to separate them. So for now, you want to keep them together in a bundled package. So here you can see you've got maybe dad 1 over here, mom 1 over here. They go through replication. You can see now you have your 2 sister chromatids. You have 1, 2, 3, and 4. And then what you have to do is you have to separate these sister chromatids. Now notice what's happened now. In a cell with 46 chromosomes, how many chromosomes do I have now after replication? So in replication, you double everything, right? So I have what? From 46 to 92, right? So that's actually how you produce 2 daughter cells that are identical. Because remember, you don't want to have your chromosomes. You want your daughter cells to have 46 chromosomes as well, okay? So the only way you can do that is by doubling everything. So it goes from 46 to 92. And then you separate those into two separate piles. So it goes from 92 back down to 46. And then you have your identical pile of 46. That's how you maintain your chromosomal number without either having it or losing or getting too many or too little chromosomes. And remember, having too many chromosomes or too few chromosomes is lethal in humans. Not obviously in plants, because we've got octoploid strawberries, right? But in us, like, I mean, you guys remember your chromosome squash, right? Some of you guys had extra 13, some of you guys had extra 18, some of you guys had extra 21, some of you had an extra X, some of you had XO, Turner syndrome. But for the most part, when you're looking at the autosomes, those are the lethal ones, right? We can have a little bit of tolerance for the sex chromosomes. So like an XXY person is fine. An XO Turner syndrome person is fine. I mean, they're not fine. They're not normal, but I mean, they're viable. I mean, they're going to live. So you're going to be able to raise them as your kid. And they're going to have a life, and it's going to be, you know, there's going to be a lot of quality of life there, right? For most others, they don't even make it to term. If you have an extra chromosome, that's almost absolutely fatal. Matter of fact, it's so fatal that it's like a catastrophic fail in development. As a matter of fact, that usually leads to spontaneous abortion, usually in the first trimester. And spontaneous abortion simply is just the fancy technical term for a miscarriage. So the majority of miscarriages, the most of which happen in the first trimester, because that's when all those critical decisions get laid down in development, like where's your head, where's your tail, where's your front, where's your back. A lot of those critical decisions are made then. And if you have an extra chromosome, or if you're missing a chromosome, that's almost always an absolute catastrophic fail. So it becomes such a train wreck developmentally that you just terminate in that first trimester. Down syndrome is really the only truly viable trisomy. And even then, a Down syndrome, this is one thing they don't really tell you when you see things like, gosh, what was that? That show that was in the 90s with the Down syndrome actor. I forgot what it was, but as a person with Down syndrome, their life expectancy is maybe in their 50s. So they're not going to live a full life. You're not going to see an 80-year-old Down syndrome usually. But, was there a question? Yeah, it's a dosage thing. So most of our chromosomes and most of us were very dosage sensitive. So like when we have our two autosomes, you have two of the same genes on the maternal and the paternal chromosome. When those genes are firing, especially like in development, your requirement is to have both of those genes firing at full steam. If you have one of those missing, then your dosage is off. And so you don't have enough of that protein to accomplish your task. And that becomes a catastrophic fail. When you take a look at missing entire chromosomes, you've got many genes, not just one, many genes along that chromosome that are the same thing. So many of our genes are what's called dosage sensitive. If we get too much, it's lethal. If we get too little, it's lethal. We have to have exactly the right amount. And that's a mammal thing. Like I said, plants have a much more flexibility for that, right? I mean, they can go from a diploid to an octoploid for crying out loud. We can't even handle one extra chromosome. Forget about giving us a full set of everything like 8 times over. So eukaryotic replication is obviously, we've talked a little bit about replication, right? We've talked a little bit about some of these things, but when we talk about duplication and segregation or separation, one thing we need to understand is that this all exists within the context of what's called the cell cycle. So the cell cycle is sort of like the cellular life cycle. So in order to understand what's going on, it's helpful to understand all the events that are happening in the cycle itself, right? And think about it. If somebody was studying you and they want to understand how things are going on in your life, where you are in your age and what your circumstances are, one of the best places for them to start is to understand better what the general overall human life cycle is, right? So when you're born, what's life like? What are you going through? Like when you're a teenager, what is it like when you're going through? When you're a young adult, what is it like when you're going through? When you're middle-aged, what is it like that you're going through? When you're getting older, what is it like when you're going through? So understanding the entire breadth of the human life cycle will help you to be able to better understand where you specifically are within the entire panorama of the whole life cycle itself. So when we're looking at cell division, we're not just looking at a process, we're looking at an entire life cycle of the cell. So to really understand what cells are trying to do here, we have to understand their entire cellular life cycle. And that gets us to the cell cycle situation. This is kind of what we covered really quickly in lab. I'm going to take a little bit more time. Oh, you got a couple weeks for that. So this is basically, we start off with the first phase, which is G1. It's a gap phase, so it's a pause in the cell cycle itself. Yeah. Oh. What? That one. So let me just kind of describe where these terms come from, right? So we have these things like G1, right? Gap one, things like that. And a lot of times students will be like, okay, what gap? What are you talking about? Like, why would you call it a gap? I mean, if we know what's going on there, why would you call it a gap? This is how this came down. So when all the cell biologists were studying mitosis, what they would do is basically sit under a microscope all day long and just watch cells. Tedious work. It's definitely not for the ADD person, right? So, but what they saw was the cell doing stuff, right? Like maybe you'd see it go into anaphase. It's like, oh, hey, look, it's going to anaphase. It's really cool, right? And then all of a sudden, it would just like stop, and it would look like it would do nothing, and it would do something else again, and it would do nothing again. And so these periods of time where it seemed like it wasn't doing anything, they called gaps or pauses in the cell cycle. And unfortunately, it just stuck. We have no other better way to describe it. That's just what kind of came out. The first one of these is G1. We know what happens in G1 now. This is the growth phase of the cell. So this is your cellular childhood. So just like us, right, in our childhood, the one major task in our childhood is that we're growing up, right? We're getting bigger, just like the cell here is going from a small cell, which is typical of a newly divided cell, they're really small, and it's going to start to grow and swell to a large full-size adult cell. And that's what's happening during the gap phase. It's kind of growing, can take a long time depending on the cell type. But just like with our childhood, as we get older, we get to sort of what we refer to as our adult body. I'm not talking about like 20s. I'm talking about like teenage, right? Your years, your reproductive years, which starts basically pretty much as soon as puberty hits, right? So that's kind of like your body saying, okay, we're ready now. Let's go, right? So, but you always, just like with us, right? We kind of get to this sort of level of a body where we're reproductively ready now. But we have a decision to make, don't we? And that decision in G1 is what's referred to as the restriction point. So the restriction point, a cell has to answer a question. It's a simple question. Do I divide? That's the question you're answering. There's only two answers. It's either yes or it's no. There is nothing in between. You can't opt out of this one. You can't choose not to answer. To not answer is to answer, right? There is only a yes or a no. We run into the same decision, don't we? What's the question we're answering? Do I divide? Yeah, right. Do I have kids? And the answer is, you only have two answers to that one, don't you? It's either yes or it's no. There's no in between. And not only that, but there's a sense of commitment to that, isn't it? The reason why that's a daunting question is that that's like a scarier commitment, actually, than even getting married, right? Which is already kind of a little daunting in itself, especially for us commitment phobes, right? But if you think about it, this is the level of commitment to this is actually kind of over the top. Think about it for a second. If you decide to have kids, then for the rest of your life, it becomes your identity. You always identify yourself as a parent, not necessarily a good one, but a parent, right? And not only that, but it doesn't matter what happens to your kids, right? Whether you lose your kids or not, whether your kids are fine, parents who've lost their kids don't all of a sudden say, oh yeah, I'm not a parent. It's like they're parents still, even if their kids have been gone for a while. It's like, no, I'm a parent. I just lost my son, right? But they don't ever self-identify as not be, oh yeah, I'm not a parent anymore. Like, no, you are a parent, right? It's like a permanent identification switch. So you commit to it. The only way that that changes really is if you get promoted to being a grandparent, right? Which, I don't know. I hear it's a promotion. I'm looking forward to it, but I have to figure out the whole sawed-off shotgun thing first, because I realized that I can't shoot all my daughter's boyfriends and to be a grandpa at the same time. I realize there's a problem with that. Yeah, I'm not quite sure how I'm going to work that out just yet, but I know I got work to do there. Yeah, right. So it's like, okay, I got to figure that one out. So yeah, I'm a mess. But anyway, but it's a commitment, right? Just like this with the cell. If you decide to divide, you're committing to it. So you're going to commit to the cell cycle. No, well, that's legit, right? I mean, like, especially for a cell, I would like to use the example of the liver cell, because liver cells are partially regenerative. Unless it's a partially, it's not a strategy. So don't go trashing your liver. You'll still need to get on an organ donor list if that's the case. We'll see why here in a couple of slides. But like for a liver cell, like when you're kind of going through your G1 and you get to this restriction stage, you look around, you're like, okay, do I need to divide? Do we need more liver cells? And your liver and your communicators that were making these decisions be like, yeah, you know what? We're good. We're good. Your liver tissue's healthy. It's looking good. Don't need to divide. You just do your thing, right? And then you say, no, okay, I'm not going to divide. So in this case, basically you drop into a functional state. We call that state G0. So for like a liver cell, this would be like, okay, you go do your thing. You know, you just do what a liver cell does, right? Do your metabolism and all that sort of stuff. And that's what you do. And for us, we have the same thing, don't we? So some of us are like, yeah, no, I'm not going to have kids. Sorry. Me and kids like oil and water, right? I'd rather spontaneously combust than have kids around me. Not going to happen.
Is that G0?
G0, yes. Right. So for that person, it's like, no way in the world am I ever going to have kids at all, ever. Right. I'll strangle them before I have them. But if you do that, didn't that mean you'd be strangling somebody else's kids? That's usually frowned upon. So you'll probably go to jail for that one. Just saying. But yeah, for you, it's just like, okay, that's fine. I mean, you don't have to have kids if you don't want to have kids. And, you know, you go, you do your career, you do whatever you do, you live your life, you do your thing, and you're a productive member of society, and that's your functional state. You kind of do your thing, right? So we do the same thing. Yes, right. So I mean, and not only that, but if you go into your G0 state, it's not forever, right? So you can actually toggle back. So, and we also have that too, don't we? Because we have some people who are like, you know what? I don't really want to have kids right now. I'm like only 20. I want to go out and do some things first. I want to get my education, maybe get a bit of a career going, and maybe I'll think about it in my 30s or something like that, right? So that's kind of a situation where initially you say no, you drop into your functional state, but then a little later on down the road, it's like, you know what? I mean, I've got a little bit of career, I've had some success. I think, you know, I want to have some kids now. Granted, I mean, if you're in your 30s, you better be moving, because time's not on your side at that point. It's just saying that's just the reality of biology, unfortunately. I've always thought it was a cruel kind of a thing for nature, for guys like Anthony Quinn, famous actor, to be able to have kids when he's 60. But you take a woman who's 60, and it's like it shuts down. It's like, that's a little cruel, but there's actually very good reasons for that, just because of the way females handle their eggs, and and males handle the sperm. It's just a different biological reality. That's what that is. It's not sexism. It's just the way our biology works. Females do things very differently than males. So let's keep it interesting, right? So you're going to commit to the cell cycle now, and so you're going to move forward. The first step is duplicate, right? So that basically means you're going to be going into replication. So you're going to be duplicating your DNA. And we have an entire chapter 14, that's going to cover that. So we'll talk about that in more detail at that particular time. And then the next phase, you hit the next phase, which is gap 2. There you go. So gap 2, you're doing a couple of things. First of all, you're duplicating now your organelles, right? So remember, you have to divide everything up. So you duplicate your organelles, you start building your microtubules, because your microtubules are what's going to be moving all the stuff around. Here you will also be doing DNA repair, because you almost invariably will make mistakes during DNA replication. So you got to clean that up a little bit. And you're just generally kind of getting ready, right? So this is your last pause, your last chance to get all your ducks in a row, to get everything set up just the way you want it before you commit to actually separating. Because once you separate, there's no going back. You can't be like, oh, wait, I didn't know. Can we put this back together? Can I do this again? No, sorry. Once you separate, for good or for bad, that's what you got to live with. Okay, so you want to try to make it as good as you can. Then, of course, after G2, we move on to the next phase of cell cycle. But the 3 of these guys taken together is our interphase. Right? So that is basically the phase that's intervening between 2 mitotic events. That's what interphase means. And then we're going to get into mitosis, which is the division of the nucleus. There's 5 different phases to that. We'll talk more detail about those in just a second. And then cytokinesis, which is the division of the cell, and the cytoplasm, and the organelles. So this is the rest of it. So you start off with the separation of your nucleus, and then you separate the rest of the cell in that order. And then after this, you get 2 new little cells that then start their own G1. And here we go again. It sends all inside the cell. Yeah, the nucleus never leaves the cell. For the nucleus? Now we'll see actually how the nucleus divides when we go through the subdivisions of mitosis, which is a big chunk of this chapter, actually. Okay, so questions about how long does this take? This is always a question. The answer is fairly unsatisfying. The answer is it depends. It depends on the organism, and it depends on the cell within the organism. So for instance, if you were to take a look at fruit fly embryos, it's cheating a little bit, right? Because embryos are going to be highly mitotic. They're going to be moving quickly because they're trying to build a little fly body. First of all, going toward the larva. So they're building the larval body, first of all. So they got to get there quick, right? For them, their cell division time is 8 minutes. That's pretty quick. So just within the context of us having taught from the beginning to now, we probably would have already developed and doubled many, many times over. Now, what about mammals? Mammals on average will take about 24 hours. It depends on the cell type. So for instance, skin cells, which you shed maybe every 14-ish or so days, are actually fairly highly, they move pretty quickly, right? Because you're constantly replacing them. A liver cell, on the other hand, can take more than a year, which is the reason why if you have plans on trashing your liver and getting it all good and cirrhotic, and your plan is to just basically divide more liver cells to replace the old scar tissue that you just trashed your liver with, it's not going to work, because you're going to die of liver failure before you can actually produce enough cells to replace the damaged tissue. This is the reason why liver regeneration is not a strategy for treating cirrhotic liver. It's only organ donation. So you already need a liver ready to go.
If you wanted to detoxify your liver, would it take a year?
So detoxifying your liver is actually has more to the things that you eat and you actually filter through your liver rather than actually making more cells.
Like the liver cells that hold on to the sugar and stuff, that's not really.
No, most detoxification is just like you're consuming things that are liver healthy and that help strengthen your liver and make metabolism easier and more efficient. That's liver detoxification. Or eliminating things that are bad for the liver and that have a negative effect on the liver. That's mostly liver detoxification. Adding new cells is going to take forever. So that's not really all that much of a help for the most part. Because nobody's going to be sticking with the detoxification regimen for a year, probably. I mean, unless they're like really committed to it. But then in that case, you don't have to worry about it because you probably already have a pretty healthy lifestyle. So it's probably already a pretty liver healthy lifestyle already.
Yeah, so as long as you're not killing, it's once the cells die, that's the
Well, and with cirrhosis, it's not so much about cell death as much about scar tissue development. That's what happens. So a cirrhotic liver is actually where the cells are being replaced by connective tissue. That's what a scar is. And that creates disconnections between the normal functional collaborations between cells. And that starts to cause problems. So the more scar tissue you have in there, the more disruptive it is to the liver unit working together as a team. And it starts to create problems with it. And that's the reason why if you start getting a really strongly cirrhotic liver, you're looking at organ donation at that point. Nice. Yeah. Get a little bit of evil, a little bit of good, and you're right down the middle, right? So assuming the evil doesn't kill you, the good's not going to do anything for a corpse, but the broccoli won't do anything if you're dead. Yeah. A line and then chase it with the broccoli. Okay. I don't think I'll experiment with that one. If you know, that's okay. Not that I've heard of. Most of that is usually associated with your epidermal and your dermal connective tissue. The homeostasis is associated with that connective tissue replacement. It's an imbalance of your fibroblasts, which starts to abnormally lay down connective tissue and they don't stop. And that's because they're overactive and you don't have fibroblasts in your organs. So in terms of time frame, you guys kind of did this with your differential count, right? So you kind of counted your 50 and you sort of broke that up and you sort of determined like roughly how much time we were spending in each one. So you can imagine then that most of your time is going to be spent in your interphase, right? So your G1, G2S phases, that's where most of it's going to be spending. And then mitosis itself is actually fairly short. So it's like an hour. And the reason why this is so short is for a very good reason. Because we break down the nuclear envelope, right? And breaking down the nuclear envelope it does say what you think it says. So breaking down your nuclear envelope is like a cellular pants drop in public. Think about it. You're in the middle of a crowd. There's no restrooms. You've got to go. So guess what you have? You got to drop the pants. And you don't have anywhere to hide. There's nowhere to hide. There's no shelter. Yeah, well, you'll probably be going a little bit more native on this one, right? So just hope it's not a mess. But anyway, We don't, that's a little too much information. Let's hope it's not one of those ones that requires a fire hose. But anyway. Or a car wash. Sorry, I'm a little squirrely. This is what happens, when you're delusional for the better part of 48 hours. You're just now starting to get normal and you're still halfway delusional. But anyway, right. So imagine, right, you're in a crowd of people. You don't have a choice. This is not going to go well. Some of us have been there, right? We're like, crap. No pun intended. Crap. This is not going to go well. So the real question is, how can I minimize the absolute humility? This is one that I'm never going to undo, right? So how do you do this? Well, think about it. You got a whole bunch of people around you, right? Before you do the deed, you spend a lot of time prepping it, don't you? You're like, I don't think I'm gonna do anything like next to this guy over here because the way he kind of looks like a pedophile. So I might not do that one over there. I'm gonna move over here. These people look a little nicer. Like maybe they're a little more forgiving and they kind of understand things a little bit more, right? And so it's like you kind of spend a lot of time just trying to scope it all out, prep it all up, right? And just kind of just make sure it's just right, as good as you can get it, right? You know it's not going to be great, but you're trying to minimize as much damage as possible, right? So that's your G1, G2S phase. So you're spending a lot of time in those prep phases, getting yourself ready. But then when the moment comes for you to drop your pants, nothing has ever happened faster in your life, right? And you're like, maybe this is not going to be like, it's just going to be like an abbreviated version, right? Just enough to kind of keep the body happy for a while so I can get in a more secure space, right? We're going to just, as quickly as possible, drop the pants, do your business, whatever that may be, and then get those up fast, right? If you're not looking carefully, you may miss it. It's going so fast. Why? Why is it so fast? Why don't you just sort of sit there, grab a newspaper, and just kind of like, what? You're extremely vulnerable, right? Because I didn't say what kind of people are in the crowd. Some of these people may be your enemies. And all of a sudden now, you're like, oh crap, if I've got my pants down around my ankles, I'm a sitting duck. So what do? You move quickly. So when you're in mitosis, because you drop that nuclear envelope, that's like a cellular pants drop. That is your most vulnerable state. Your most precious commodity is your DNA. It is all now out there for everybody to see. So you move quickly, right? It could be anything. No, nothing is going to attack it because those cells are in our body. But still, the idea is you don't linger, right? Because you're in a generally vulnerable state. There's no protection at all. So if something did want to come and get you, you're there for the picking, right? You're just a sitting duck. That's the reason why the mitosis event actually is going so quickly, is because we chose to break down the nuclear envelope as opposed to keep it intact like protists do or yeast does. It requires us to move very quickly because nobody wants to have their pants down in public. I mean, unless you're like kind of into that sort of thing, and that's a different class, probably more like psychology, maybe. Can't imagine which one, but is that what it'd be? Human sexuality? There's probably a name for that. I don't know what it'd be, but it's kind of the opposite of voyeurism, right? Like exhibitionism. That's it. Yeah. So, yeah. Well, it's interesting. Right? Yeah, you're like, it's not because it's like, yeah, I learned all this stuff. I'm like, kidding. Yeah, you're all like. So that's basically your cell cycle, right? And of course you have your G0. So at this point, you're kind of in your resting state. You're just kind of doing your thing. And you can stay there for as long as you want. So it's kind of like your cell cycle. So here you can see this big old long G1, right? You spend a lot of time there in G1 growing up. And then you commit to the cycle, you go into your replication phase, your S phase, you go into your G2, which is your preparation phase. And then you hit your mitosis phase. That's this big purple one here. So you can see it's broken up into several subdivisions. Prophase, prometaphase, metaphase, anaphase, telophase, right? We learned about all those in lab. And then, of course, you follow that up with cytokinesis. So that's going to be this phase right here. And around you go again and again and again and again for as long as you're doing mitosis. So we have our interphase and then the rest of it, right? And so a couple of things just to sort of clean up when you're taking a look at interphase. One of the things that happens actually in interphase, I mean, we already know about G1, but in G2 in particular, right? We already know that you have organelles replicating, including the centrioles, right? Those are important because you're going to be building your spindle. You have tubulin synthesis, so this is protein synthesis. So this basically means that you have lots of ribosome busy making this tubulin so you can build these microtubules with, right? And then what else goes with movement? Your motor proteins. Remember we talked about dynein? So you got to make those too. So protein synthesis has to make these motor proteins as well. So in G2, you're not just doing a couple of things. There's a lot of things you're doing. So you're trying to lay the groundwork and lay the materials down for everything that you're going to need in order to execute this movement. And there's a huge network, right? Because right now, as it sits, the network is not in place. You don't have microtubule tracks that lead you to where you want to go. You have to do a lot of road building, which means you got to secure all the asphalt or the concrete. You got to do grading, right? And so you got to do smoothing and all the road prep stuff before you actually lay down asphalt. And you generally don't lay down asphalt on dirt. You usually lay concrete down there first and then asphalt on top of concrete, right? And so then you have to do all that. So you got to make sure you have enough asphalt, enough concrete. You have to have enough machines to actually do the work. You have enough labor to do the work, and then enough manpower to do it. Those are the motor proteins, the actual yellow jackets to actually execute the job and actually do the job of the road building. And that's before you even get to the thing you're going to move in the first place, right? Which is the chromosome. So while you're doing this G2 stuff, you're doing a lot of stuff in there. So a lot of the chromosomes at this particular point in G2 are also condensing. So we talked about chromosome condensation. So in G2, chromosome condensation begins. Remember, packing down your fine china begins in G2. So you're starting to pack all this stuff up, along with making motor proteins for movement, making tubulin to build your microtubules, laying down the centrioles to be able to develop, to be able to sort of build that spindle, and then also, making more mitochondria. So you're duplicating all the rest of the organelles as well. And then on the chromosome side of things, you're also doing a lot of work. So you're building your kinetochores, which are little protein pads at the centromere for you to grab onto, right? Because you never want to just grab onto DNA because it'll shred. So you build this little protein handle, if you will, right there in the centromere that allows your microtubules a place to attach and to be able to pull those chromosomes without having to shred them or damage them. So all of these then are basically happening in G2. So you have lots and lots of things going on in G2. It is an incredibly busy time in the cell. So it's kind of ironic that they call it a gap, right? This pause where it looked like nothing was happening, when in reality, there's a ton of stuff happening. Ultimately, this is kind of what it's going to start to look like. So you can see here's your chromosome. You can see this is post-replication, right? Because you can see your two sisters here. Here's one of them. Here's the second one. They're held together in the middle by that glue-like protein, cohesin. And you can see your little protein pad there, the kinetochore on the centromere. And then here they kind of attach some microtubules to it. So you can see how the microtubules will attach that kinetochore. And all they do is pull them in opposite directions, kind of like a tug of war. And as long as your cohesin lets go, your sisters will pull apart. And that's kind of the basic thing, the basic gist of where you're going with this. So let's take a look at some of this. First, we're going to start off with a G2 interface. Notice what's happening. First of all, homogeneous, right? That's how you know it's interface. So you don't know if it's double or not because you need the reference point for that. But you can see you've got nucleoli here. That means you're actively doing protein synthesis. Why? Because we just said we're making motor proteins and we're making lots of tubulins. So you can expect that. But also you see here, we've got the centrioles duplicate. And the chromosomes are condensing. So it starts here. The spindle poles of the centrioles duplicate. As well as other organelles. You have protein synthesis going on. And this is your last chance to get it right. DNA repair. Make any mistakes in your DNA, you gotta fix those. Right? Because think about it. You can't tolerate any mistakes in DNA. Your DNA has to be a perfect copy from round of division to round of division. So there's like a whole spectrum of different DNA repair mechanisms of different types, many different types. And they start actually during S phase, and they're continuing to be active into G2. And so there's lots of different types of repair mechanisms, and they're looking for very specific types of mistakes. Like some are mismatch repair, where you just simply mismatch a base pair. Like instead of an AT pairing, you put like an AC pairing, right, which would be a mismatch. So there's mismatch repairs. There's what's called excision repairs, where they look for like sort of structural anomalies in the DNA. They just cut it out and they just relay it back down. There's like all kinds of different repair mechanisms, and they're all very active. We have a lot of them, actually. They're mostly protein. Yeah, they're mostly like they're like either protein complexes or they're a series of protein complexes that kind of create like a pathway effect, like a step 1, step 2, step 3. Like it's not uncommon to see like one protein complex whose job it is to recognize the damaged DNA, and then it'll recruit in another complex in to cut the DNA out, and then it'll recruit another one in to actually lay down the new DNA and to fix it. So there's like multiple protein complexes that sort of collaborate together. And that's a pretty common thing that you see that. And we do a pretty good job of fixing a lot of it. And there's a lot. And if you think about it, it's actually kind of amazing how good it is, right? Because think about, since you were a single cell, how many rounds of mitosis have you gone through to where you are right now? 2? 3? 100? 100,000? Millions? Trillions? And think about it. Every single time, the 2 daughter cells have to be identical. No slip up. It's better than a Xerox reproduction, right? Have we all done the Xerox reproduction game? A copy of the copy of the copy of the copy, and then see just how bad the 10th or 12th copy is, right? We all done that one. Or we've heard of it done, because it's harder to do these days, but because we're all digital, right? But it was a lot of fun, because like within about 5 rounds of the copies, it's like, what is this thing for? It's like everything's all filled in. It's like the E's are filled in, like they're all just blobs all over the place. It was a lot of fun, right? But the idea is, not only do you have to minimize that, but you have it has to be like a 0 fail mission. It has to be like a 0 error mission. Think about how difficult that is. We don't have anything like it in human existence, where something over so many generations is completely immaculate. And it has to be that way, right? Because let's say that each mitotic division had a very low error rate. Let's say it had maybe 1%. If you think about that, by the time you built your trillions of cells of your body, you'd be a train wreck. Because that 1% would compound from generation to generation to generation. So by the time you got to trillions of cells, those cells would be so screwed up that they would be functionless. So being able to actually have this level of perfection is actually a pretty amazing thing.
The red.
This is part of the spindle. So that's why I saw a cytoskeleton they're showing there. Probably microtubules, I guess. So they're kind of showing how the microtubules are starting to grow and expand.
Is that what the asters are?
Yes. Yeah, the asters, which I refer to as spindle poles, a couple of names to them. Asters, because of the way they look like asters. But my experience is most students don't know what an aster looks like, because or what an aster is. How many of you know what an aster is? Like a flower, right? But a lot of students have no clue what an aster actually looks like. So it's kind of meaningless for us to call them asters, which is the reason why I tend not to call them asters. I call them spindle poles, which makes sense, right? Because we're building a microtubule spindle around the nucleation point, which is the pole, right? That's the reason why I call them spindle poles. So then we're going to move into mitosis, right? And you have 5 different phases. You have prophase, prometaphase, anaphase, telophase, anaphase, and telophase. Now, when you're looking at cell cycle, oftentimes you'll have 2 different camps. You'll have the 5-step folks and you'll have the 4-step folks. Like me, for instance, I'm a 4-step person. And the phase that's in question is this one right here. Now, it's not wrong, right? The cell cycle is what it is. It's a continuum. It's all a matter of where we draw lines and how we hang names. That's pretty much all it is. But typically, this can be a point of confusion, is because when we get into meiosis, you almost never hear the word prometaphase again. And a lot of times when you're looking at meiosis, like you would have a prophase 1, a metaphase 1, an anaphase 1, a telophase 1, and you never hear prometaphase again. And a lot of times that confuses students because they're like, well, wait. What happened to prometaphase? Is there a prometaphase here? Well, there is. You're still doing the same things. It's just that meiosis people tend to see it differently. So, and I'm from a meiotic lab from UC Davis. So that's how I learned it as a 4-step system. So let me tell you where it comes from. If you see prometaphase, typically this comes from somebody who is largely a microtubule biochemist, because a lot of what's happening in this particular area is a lot of microtubule dynamics that are going on in the arrangement and organization of the chromosomes. Okay, that's what's happening in prometaphase. And so there's a lot of microtubule work going on there, which is the reason why the microtubule biochemists really like this phase and they want their own phase, so they call it prometaphase. In meiosis, we don't care. Because in meiosis, you're usually studying meiosis and you're usually studying genetics. So that's what you're doing. So you don't care about the microtubule dynamics. That doesn't mean anything to you. So you tend to see the world as prophase, metaphase, anaphase, telophase. And then what you do is instead of creating a prometaphase, you subdivide prophase into subdivisions. So like there's different stages of prophase. Like there's silly names like pachytene and zygotene and diploatine and leptotene. If you hear those terms, you're talking to a meiotic person. It's all the same. The cell doesn't care what we're calling it, right? It's all doing the same thing. It's just a matter of just our view of the world, and the biochemists throw in a prometaphase because that's what they geek out on, is their microtubules. And then the meiotic people just focus on the development of the homologs, the chromosomes, as they're shifting around. That's the reason why they talk about the other 4 phases, and they don't really talk about prometaphase. So when we get to chapter 11, don't be confused. It's just a matter of perception. Okay. Now, oftentimes when I cover it, I also skip over prometaphase because I like to explain prometaphase as a transition from prophase to metaphase, as opposed to its own phase in its own right. So let's start off with prophase. Let's see what's going on here. So first of all, number one is in prophase, your chromosome condensation continues and completes. So because of this, remember it started in G2, it crosses over into prophase and it completes. This is the reason why when you look at a prophase cell, if you remember from the mitosis lab, they start to look like gummy worms. They start to look like compacted little night crawlers with a lot of space in between them. It's because of that condensation that's happening. And the other big thing that's happening is spindle migration and assembly. So remember your spindle is duplicated in G2. In prophase, they are migrating into their antipolar position. And then they'll start to build out their microtubular ring. And start building microtubules off of them. And then the other big one that happens is your nuclear envelope breaks down. That's also happening in prophase as well. Now, they're not all happening at the same time. They happen, some of them happen in early prophase, some in middle prophase, some in later prophase. So there's like a spectrum of early, middle, and late prophase. That's the reason why meiotic people break prophase up into these subdivisions, like liptotine and diplotine and zygotine and pacotine and things like that. But those are the main things that are happening in prophase. So here's your prophase cell. So you can see your spindle poles are migrating into their antipolar position. You've got good condensation now with lots of space in between them. So here you can see also your nuclear envelope is now gone. So you can see there's no more boundary around those chromosomes up there. They're just, and they're like the little night crawlers, the gummy worm. So you can see lots of space in there, absence of a nuclear envelope. And you can see a nice clear antipolar position here and here as you're building your spindle. So that's prophase. But let's take a look at where we're going first and we'll see how we get there. Okay. Metaphase is the next stop. So with metaphase, what's going to happen is you have a well-developed spindle pole and all of your chromosomes are now attached to the sisters at the kinetochores. And everything is lined up in the middle with this little imaginary line called the metaphase plate. So this is your line of central tendency that we talked about in lab. So here you can see your metaphase plate. It's going to be lined up here at the center. That's basically your metaphase. So everything that is on one side is going to be moving toward this pole. Everything on this side is going to be moving toward this pole. And the metaphase plate sort of creates a line of division. Now, how do you get there? Right? How do you get from this to this? Well, the process of getting your chromosomes lined up is what's happening in prometaphase. And the reason why we're interested in this is because notice what's happening. The first thing that happens is your chromosomes kind of progressively just sort of fall into mush. And you're actively trying to grab onto them and you're trying to sort them all out. So here you can see you've got one spindle pole that's grabbed onto a sister over here. The other one's grabbed onto it over here, but they're not lined up yet. So what happens is during prometaphase, it's kind of like a fly fishing mechanism. I don't know if you've ever seen somebody fly fishing. Not a fisherman myself, but I watch it on TV. Not very successfully. I've watched a river run through it a couple of times. So I'm an expert now. I know how to fly fish. I know just enough to not do it. Anyway, right. So if you take a look at a fly fisherman, they're like casting out, right? And they're kind of pulling back, casting out, pulling back, casting out, pulling back. And so what the spindle pole does is it basically builds microtubules and it kind of casts those microtubules out. And it's what it's doing is it's trying to lock onto one of those kinetochores. And in some cases, it won't find it. And so what it does is it breaks it all back down, pulls it back in, and it'll cast out in another direction. And then it won't find it again, it'll break it all down, and it'll cast out in another direction. And then eventually what's going to happen is it's going to cast out, it's going to find a kinetochore, and it's going to lock onto it. Then what it does is it waits for the sister pole to grab onto the other side. And once they do, then they kind of tug on each other and they kind of kind of shift it back and forth so they can say like, okay, it's a little too close to me. Why don't you pull a little harder and pull in your direction? So we got to get this thing lined up here in the middle. And then they kind of are constantly tugging on each other. They're feeling each other's tension and they're kind of getting that slowly kind of all lined up in the middle. And there's a bit of a process associated with that, right? So it's just like a fly fisherman. I mean, if you know what a fly fisherman is doing, it kind of seems like, well, why don't you just cast it out there, right? A bait fisherman is different, right? If a bait fisherman is doing that, they don't know what they're doing, right? But a fly fisherman, it's a different technique altogether, right? It's not that they don't know what they're doing. What they're doing is they're messing with the fish. The idea of fly fishing rather is to tempt the fish to grab your lure, because you're trying to get the fish to believe that your lure is some sort of a fly skittering across the surface of the water. And that's the reason why you cast it and you're sort of touching and teasing the top of the water, trying to get them to strike. So sometimes it works and sometimes it doesn't. And sometimes they're interested and sometimes they're not, but you keep going back and forth. You keep kind of teasing that water a little bit with your lure. And if they like what they see, then they'll jump at it, grab it. And as soon as you got it, you've got your fish. Like I said, I know just enough to not do it. The one thing I like about the fly fishing idea, though, is that even though I'm not much of a fisherman, of all the fishing strategies, that's probably the one I have the most respect for, because there's a lot of strategy in fly fishing. It's a thinking man's fishing game, right? I mean, anybody can drop a stick of dynamite in the lake and get fish. That's kind of like bait fishing, right? That's kind of cheating. But fly fishing, I mean, you're in moving water, so you got to really think like a fish. I mean, that's one of the things I like about fly fishing. It's very strategic. It's very, and it changes from day to day. What worked yesterday could be completely different than what's going to work today. So that's kind of what's happening. And then eventually what happens is you slowly lock onto all the chromosomes and you kind of shift them back and forth. And eventually you get them all lined up in the middle and you find yourself at metaphase. But wouldn't we like to talk about anaphase? We'll have to wait. Suspense, right? It's killing you. You have to wait till Monday. What? This? Yeah, it's kind of like a lined up in single file. So it'll basically kind of line itself up. And then what's going to happen is it basically just pulls them apart. Yeah, so 3-dimensional. Yeah, so there's 3 dimensions. Yeah.